Ocrelizumab is already a cornerstone therapy for multiple sclerosis, but many patients still worsen. Could a higher dose finally stop disability progression? Here is the paper itself. Before these trials, the approved 600 mg dose was proven to cut relapse rates and delay disability. Yet, disability that occurs independent of relapses remains a major unmet need, and earlier analyses hinted that higher drug exposure could be more protective. Both studies used 12-week composite confirmed disability progression, or CCDP, as the primary endpoint. It captures any meaningful worsening in walking, hand dexterity, or overall disability. Hauser, Giovannoni, and colleagues asked whether a higher, weight-adjusted dose of ocrelizumab could further reduce disability progression in both relapsing and primary progressive MS, without adding new safety concerns. The MUSETTE trial enrolled 860 RMS patients, while GAVOTTE enrolled 753 PPMS patients. Participants were assigned in a 2-to-1 ratio to a body weight adjusted high dose or the approved 600 mg dose, and received infusions every 24 weeks for at least 120 weeks. Here you can see the flow of participants. In MUSETTE, 577 received high dose and 283 received 600 mg. In GAVOTTE, 500 received high dose and 253 received 600 mg. Follow-up continued until a pre-specified number of disability events occurred. Disability was captured using the expanded disability status scale, walking speed, and hand dexterity tests. MRI evaluated lesion activity, and safety was tracked through standard adverse event coding and laboratory monitoring. In the relapsing MS trial, the high dose yielded a hazard ratio of 0.93 for 12-week CCDP compared with standard dosing, a difference that was not statistically significant. In the primary progressive cohort, the hazard ratio was 0.95, again showing no meaningful advantage for the higher dose. These survival curves illustrate that the high dose and standard dose arms overlapped closely in both trials, confirming the lack of a significant difference in disability progression. Safety profiles were essentially identical. In MUSETTE, 96% of high-dose participants reported any adverse event versus 94% in the standard group. Serious events and deaths were also comparable. GAVOTTE showed the same pattern. Even though the high dose achieved deeper B-cell depletion, it did not improve clinical outcomes. This suggests that mechanisms beyond peripheral B-cell counts drive progression, and that the standard dose already provides maximal benefit. The trials were powered to detect a relatively large hazard ratio reduction, so modest benefits could have been missed. Follow-up stopped once the required number of events occurred, limiting long-term insight, and the studies did not explore CNS-resident B-cell dynamics. Future research may need to look beyond peripheral B-cell depletion, perhaps combining ocrelizumab with neuroprotective strategies or drugs that penetrate the central nervous system more effectively. In practice, continue prescribing the approved 600 mg dose. Recognize that higher dosing offers no extra advantage, and look for complementary approaches to address progression that is independent of relapses. Clinicians should avoid escalating ocrelizumab dose purely to chase deeper B-cell depletion, as the trials show no added clinical benefit. This figure demonstrates that the high-dose regimen achieved markedly lower peripheral B-cell counts, yet this pharmacodynamic advantage did not translate into better disability outcomes. The trials were robust, enrolling over 600 participants worldwide, using a double-blind, parallel group design and providing detailed safety data. We appreciate the work of Hauser, Giovannoni, Montalban, and all investigators for delivering these important trials, and thank the Lancet for publishing the findings. The take-home message is to let high-quality evidence drive our treatment choices, and to stay tuned for the next wave of discoveries that will further improve outcomes for people living with multiple sclerosis.